Okay, we are live with Otangelo, and we are waiting for Dmiter to get back from an errand, which will be about 15 minutes. And Otangelo has the God Talk channel, which I will link in my description. And let's see what else. You're going to play us a video that you did on irreducible complexity.
Yes,
I have to get rid of this. I have a problem. There we go. Problem gone. Anyway, how you doing this morning?
Fine, thanks. And you, Mike? How are you?
I'm good. So that 9-hour stream, I wanted to see parts of that, and hopefully you can get that back.
Yeah, if you, I wrote Godless Engineer this morning, and I explained to him that I didn't know about the copyright rule.
Right.
And he promised that he would remove the strike, and I don't know if after doing that the video will be put back and be available for anyone to watch.
Yeah, I didn't know about that either. I never thought about it, but actually, I guess when you're doing that, you're taking away from their stream.
Yeah, so, but I told him I had just 10 people, which are actually not his viewers. They are more circling me, so I didn't take anything away from his audience. So.
Yeah, you only have 9 viewers.
It was really an unjustified move, but okay. Let's hope that he removes the strike and that we can move on.
It won't be the first time we do something to each other that aggravates one another.
Yeah.
You know how it goes. You're getting used to it.
Yeah. Okay, so the link to my video is here in the private chat. If you want to screen it.
Oh, okay. So let's see. How can I? Let me see if I can do that. Okay, so that's a YouTube video.
Yes, it's on my channel, Intelligent Design Academy. It was one of the first videos that I made.
Okay, let me see where I can fit it. Okay, so I got the sound off right now. I got to put my headphones on, otherwise it echoes. God, you were online, what, 10 hours, 12 hours yesterday?
Yeah, something like that.
Yeah, crazy. Okay, let's see. Here we go, is photosynthesis. Okay. Do I need to make the video bigger? I'll go ahead and start. I'll go ahead and start playing it.
You can put it full screen.
Yeah, actually, I don't know how to. Oh, I know how to do that. Well, let's see.
Yeah, just click on the square symbol on the right side on down.
Oh, that works. Okay, I didn't know that worked.
Put it full screen.
Okay. So go ahead and run. Now, speak very loudly if you want me to pause it. Okay.
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 Oxygenic photosynthesis is an elegant, extremely complex biological pathway that defies evolutionary explanations, which points overwhelmingly to intelligent implementation and design. Imagine you had to install a power photovoltaic system in your house. At least 5 individual interconnected parts would be absolutely essential in order for the system to work and able to store electrical energy. At least 7 individual processing steps in a solar panel factory are necessary, and each step produces an intermediate product which by itself has no function. The production of solar panels requires complex factories and machines. In the same manner as solar panels require complex manufacturing processes, so do batteries, charge controllers, inverters, and AC service panels. Each of the individual parts of the system have to be made in specialized factories by automated robots, production and assembly lines tailored to produce the specific components of the photovoltaic system in demand. If any of the 5 components are missing, the system cannot become functional. Solar energy cannot be gained and stored. The final goal cannot be achieved and accomplished. That means this is an interdependent, reducible system where all components are essential for reaching the final goal to store energy. Evidently, no planner would invent solar panels and the respective factories and machines without the other 4 parts required for the photovoltaic system, cabling, and other minor parts without considering the final purpose of the system. The origin of blueprints, instructional information and master plans, and the manufacturing of complex machines and production systems upon these plans are both always tracked back to intelligent sources which made them with specific purposes. The photovoltaic system is a perfect analogy to describe the photosynthesis pathway, one of the most important chemical processes occurring in life. And we can draw some interesting comparisons and conclusions which will illustrate why this pathway was far more probably made by a creator of extraordinary power and intelligence rather than by Darwinian evolution. The evolutionary narrative goes that life started about 4 billion of years ago with the last universal common ancestor, and from there life forms evolved and branched into the great biological diversity that we see today. A major evolutionary event supposedly occurred about 2.5 billion years ago when cyanobacteria emerged off the scene and began filling the atmosphere with molecular oxygen through photosynthesis, called the Great Oxygenation Event, and so the primary source of energy for all life forms providing oxygen for respiration of advanced organisms. We find certain generalities regarding photosynthesis. An important one is that photosynthesis occurs in membranes. In photosynthetic prokaryotes, the photosynthetic membranes fill up the cell interior. Photosynthesis also takes place in plants, in organelles of leaves called chloroplasts. A tree, a plant, an algal cell, or a cyanobacterium. At the base of virtually every food chain on Earth is a photosynthesis performing organism. Through the process of photosynthesis, carbon dioxide from the atmosphere is fixed in the Calvin cycle using sunlight energy to make glucose sugar molecules, the primary source of energy for all life forms. Oxygen is a waste product in an early stage of the process. Product of photosynthesis, glucose, is a carbohydrate sugar needed as food source for almost all life forms. Photosynthesis should take a center stage right after the origin of life, when it comes to elucidating biological origins, since it is one of the most important chemical processes. This critical biological multi-step pathway depends on some very elaborate and rapid chemistry involving a series of enormously large protein complexes all working together in an interdependent manner. Photosynthesis consists of light-dependent reactions and light-independent reactions, in special the Calvin cycle. In plants, the so-called light reactions occur within the chloroplast thylakoid membrane. In the pathway, at least 24 protein complexes are absolutely essential and indispensable. A good part of these complexes is uniquely used in photosynthesis. The pathway must go all the way through, and all steps are required, otherwise glucose is not produced. So, photosynthesis is an interdependent system that could not have evolved, since all parts had to be in place right from the beginning. It contains many systems composed of parts that would be useless without the presence of all other necessary parts. In these systems, nothing works until all the necessary components are present and working. So how could someone rationally say the individual parts, proteins and enzymes, cofactors and assembly proteins not present in the final assemblage all happened by a series of natural events that we can call ad hoc mistakes by lateral gene transfer? As demonstrated previously, without solar panels, no solar energy could be captured, and a solar power system would be non-functional. Human-made solar panels exercise the same function as chlorophyll molecules in photosynthesis. A modern leaf with 70 million cells will contain about 5 billion chloroplasts, each containing about 600 million molecules of chlorophyll. Chloroplasts are organelles that conduct photosynthesis, where the photosynthetic pigment chlorophyll captures the energy from sunlight. Chlorophyll is the world's most abundant organic material. Without chlorophyll, no light is absorbed, and photosynthesis cannot occur, and therefore energy in the form of glucose cannot be produced, stored, and later used. Light is absorbed by chlorophylls, and the light energy is transformed into electrochemical energy and then into energy stored within chemical bonds. In the chloroplast, chlorophylls are bound to the light harvesting complex, which is embedded in the thylakoid membrane. Pyrrole rings are responsible for the color of chlorophylls and also called chromophore. The basic structure is a ring made of 4 pyrroles, a tetrapyrrole, which is also named porphyrin. Its large inner surface area has a high cross-section suited for photon capture. The tetrapyrrole system shows a high absorptivity at both the long and short wavelength ends of the visible spectrum of light. The middle of the light spectrum is not absorbed but reflected, which is the reason why leaves are usually green. How exactly do chlorophyll pigments capture the energy of light? In the picture, we see 2 photosystems which are surrounded by light harvesting antennas. Their chlorophylls are embedded, capturing photons. The photosynthesis pathway starts with light absorption, which excites the chlorophyll molecule. And then the energy is passed from molecule to molecule until it reaches the reaction center complex. Here, an excited electron from the special pair of chlorophyll A molecules are transferred to the primary electron acceptor. When chlorophyll absorbs a photon, an electron excitation in its ring structure occurs, which moves electrons from the ground state into a higher excited state in the atoms. A sequence of alternating double and single bonds in rings, which form a system of conjugated bonds, is responsible for light absorption. A photon is a very fast-moving packet of energy. When it strikes a molecule, its energy is either lost as heat or absorbed by the electrons of the molecule, boosting those electrons into a higher energy level. Whether or not the photon's energy is absorbed depends on how much energy it carries, defined by its wavelength, and on the chemical nature of the molecule it hits. Electrons occupy discrete energy levels in their orbits around atomic nuclei. To boost an electron into a different energy level requires just the right amount of energy, just as reaching the next rung on a ladder requires you to raise your foot just the right distance. Magnesium is present in the center of the ring as a central atom. If other metals instead of magnesium are used, they will competitively quench and extinguish the chlorophyll's excited state and return to the ground state. Consequently, very little, if any, energy transfer would occur. This means that insufficient energy would reach the reaction center and the process would not continue. Magnesium chlorophyll has a unique property, which is that the absorption and emission spectra overlap exceptionally well. That means that energy transfer to nearby molecules by the first resonance mechanism is very favorable. A specific atom can therefore absorb only certain photons of light, namely those that correspond to the atom's available electron energy levels. Magnesium chlorophyll has almost the best possible overlap of absorption and emission spectra of any molecules. All of this illustrates that photosynthesis is finely tuned and small changes can have dramatic effects. Copper and zinc form complexes with chlorophyll but with different affinities and functions. How could a natural selection fix the right information in the genome to instruct the import, purification, and precise insertion of the right central atom with the required functional properties?
Hello.
Oh, putting you back in the stream.
Yeah. Why stopped?
Huh?
Why you stopped?
Why I stopped?
Yeah.
Because you're here.
Ah, okay. Makes sense.
Yeah. Yeah, there's another 15 minutes, so
Yeah.
and people are clamoring for a conversation in the side chat. So
Is there any other one will come?
Anyway, let's see. I'm going to put this link in the description for anybody who wants to watch this video, Otangelo. Okay. And I'm just going to let the two of you talk, and I'm going to mute myself and try to stay out of it. Is that all right?
No, you can still be inside. I don't see why you should mute yourself.
Oh, I might comment occasionally, but I don't know. We'll see if I have something to say.
Who else will come?
Molecular Alchemy might stop in, but he's in a parking lot right now, so I'm not sure if he can talk. Immutable, and everybody knows about this.
Okay.
So I will bug them again and see if they want to come in. Okay.
Let me grab my coffee and I'll sit down.
Okay. Otangelo, how did you do that video? What software did you use? Just PowerPoint streaming? Or are you there? Oh, you're muted. Otangelo, I meant Otangelo. You're still muted.
Okay. I use VSDC software.
Used what?
To make my videos. VSDC.
VXDC.
VSDC. That's a video editor.
Wow. Okay, you guys are on. So what would you guys like to discuss? We were looking at a photosynthesis video.
Okay. So how is my voice?
It's really good.
Do you listen well? Okay. So because the microphone is here, this is kind of very cheap microphone. That's why I'm a little bit.
Yeah, you guys both need to get standalone microphones, but yours is pretty good. Yours is still a little bit low, Otangelo. I don't know why.
I don't know why either, because I have my volume fully up.
Yeah, it's pretty good today.
Is it better now?
Yeah, it's all right.
Okay.
So go ahead.
Okay, so let's start somehow slowly and carefully. First, I watched the video at Angelo with Mike, probably yesterday or whatever. And this is the reason why I requested just to meet again and discuss again for the science and the science issues. Also, what actually kind of caught my attention was that you debated with all the others, and it seems that all of your arguments, everything that you put as arguments everywhere, first of all, was the same as before, but it seems to me that all the arguments was kind of without, it's looked like that we never talked with you. It's looked like that we never discussed these issues. So I'm giving my arguments and everything. I'm explaining how the science works, what the evolution is, what is actually how the scientists thinking in advance, and how the process is going. I'm explaining, I explained to you all about the complexity, about the origin of life, about prebiotic earth, about prebiotic synthesis. Everything was discussed with us. And again, you're coming with the same arguments over and over again. So there's either you don't understand what I'm saying, or you completely disagree with what I'm saying. So this is two possibilities. Probably you don't understand what I'm saying. And everything that was kind of discussed yesterday have a very good and reasonable answer. I saw that Immutable tried to answer a few things, but he was kind of not prepared probably to directly point the argument. So I will go a little bit over a little bit by what I saw yesterday, and I'll continue to give you the perspective of the modern today's evolutionary biology.
Mike, would you like to remove the screen since we are not watching the video anymore?
Yeah. So before, I mean, I decided that probably it's a good idea before to start debating. Actually, I have a few simple questions to you. My first question is, do you think that scientists at all might come to some sort of objective reality by scientific methods?
In regards of operational science, to explain how things work, yes. In regards of his...
So what do you mean? Yeah, what mean by operational science?
Yeah, when a scientist tries to investigate the protein, how it works, then he will make his investigations. He will try to find out the architecture of the molecule, and then he will describe it, and then he will try to understand what it does, how it does it. That is operational science.
So everything that goes in a tube and goes in preparation and actually shows in front of the current experiment how it goes with, let me say, additional methods like, let me say, absorption analysis or kinetics or some sort of reactions that have input and output of reactions, all kinds of those things. You think the science is correct? Is that my understanding?
Well, it's not that it's correct. We can actually investigate. We have very advanced methods to go into that dimension and find out with high precision what occurs in the molecular world.
Okay, good. Next question.
Now, I didn't finish. That is in regards of operational science. Now, in regards of origins, where we try to explain how things came to be, that is not something that we can empirically verify. What we can do is, from what we do know, come to make reasonable inferences of how things could have originated in the past.
Okay, I understand that. So you want to say that if something happened in the past, you're not quite sure that happened in this way or another way.
I think nobody can say that with absolute certainty.
Okay, with absolute certainty. Okay, so I'll come to this point a little bit later. So my second question is, what is your understanding, and to what extent you accept the evolution, or you do not understand, or you do not accept the evolution?
Well, evolution, of course, is a very broad term which has to be defined what is meant by it, and we have different levels. We have what is commonly said to be microevolution. I would rather describe it as adaptation, where organisms need to be able to adapt to the environment and to the different conditions of the environment. And they have, in my understanding, pre-programmed mechanisms to do so, like heat shock proteins and so forth. And these are mechanisms which had actually to be already there when life started, because otherwise the organism would die. Now, another aspect of evolution is common ancestry, the claim that all life forms, they started from a universal common ancestor. This is a claim which I think there are 7 important points which disprove that claim. And one is that the DNA replication of eukaryotes and prokaryotes is different. The phospholipid membrane is different. I think we actually covered this already. And then the third claim is that from that supposed universal common ancestor, the 3 domains of life originated and then diversified, and from eukaryotes, multicellularity emerged. Now, this is, in my understanding, a claim which is not supported through evolution. I think that the traditional mechanisms proposed, which is mutations, natural selection, genetic drift, and gene flow, they are gene-centric explanations. And what we know today is that in order to build complex organisms, much more than just genes are required. I have listed 18 different mechanisms in special epigenetic mechanisms and 24 epigenetic languages, which are all involved in organismal making, in the organismal architecture, form, and development. So upon investigations, we know that natural selection has a very limited range of being able to improve the organism. I think it's just about two mutations, which would take about 100 million years to fix in the population. And what science has discovered, that evolution, rather than evolve or advance the complexity of organisms, it devolves. It is there is thermodynamics in place which actually devolves information and the genes and the genome. So I disagree with the evolutionary, the modern synthesis in regards of that question.
So basically, in a small changes, it's already predisposed everything, and it's predetermined all of the changes in a small scale, when something like a species can adapt better to some environment, but in a large scale, it's not possible. Is that?
Yeah, I think that primary. I think just quick. I think that secondary speciation, where populations separate and then cannot interbreed with each other, that is a fact. That is also macroevolution, but macroevolution of primary speciation, where we see the emergence of completely different body plans and new features like eyes and wings and foods and shells of turtles and so forth. That is the kind of evolution which I don't think is evidenced in what we observe.
Okay, so now I understand your statement about the evolution. My third question is, how old is the Earth according to your understanding?
I think it's about 6,000 to 6,500 years old.
500?
I'm a young, no, 6,000, 6,500. I'm a young Earth creationist.
Is it possible to be 7,000 years old?
Maybe, yes.
Okay.
I'm not so sure about that.
Okay. So, and my last question is, how frequently you are listening Discovery Institute scientists?
I've been at the Discovery last year. I've participated at the summer seminar there, and I've actually wrote 2 articles for them. And recently, I'm not reading much about their information. Most of them, they are old Earth creationists, which I am not agreeing with. Very few are young Earth creationists. But my information is principally secular peer-reviewed science papers and books.
Okay. So I mean, pretty much I tried to figure it out where you stand and what is your statements and things like that. So I was very surprised actually last time when I saw you debating with Mike and the immutable and all of that, that you haven't used any arguments that we discussed with you. You just simply ignored. So let's try.
I did send an email, Dmiter, to you, to Mike, to Leo, and also to the rest of the crowd, which is usually assisting our channels. And I think I pretty clearly exposed what problems that I see in your peptide hypothesis.
No, I'm not there yet. So I will come to this point eventually. But our discussion, it's not just for that. I mean, there was a fundamental biased claims which are completely wrong from you. They're completely wrong. They're not scientific claims.
Yeah,
and that's my
point them out.
and
yeah,
that's
point them out. You can discuss them.
So first of all, let's say a few things about science as a subject, as a things that are what they are. The science does not, it's never used, at least we never used a term or words like a belief. We don't believe in anything. The science, it's doing experiments, and the science do a proof or give a evidence. There is no belief in science. We don't believe that two moleculars interact in each other. We're observing that. So that's first thing you should know. The second, there was a very interesting, when you discussed yesterday, when you discussed yesterday with the other guys, there was a questions that you asked actually, what I need to see to happen, I need to see in order to be convinced that ID exists, some intelligent designer exists. That was a clear question from you to the others.
Yes, exactly.
So you asked the same.
Because in my framework, Dmiter, intelligent design should never have been excluded a priori in regards of historical sciences, because it is a possible explanation for what we observe in the natural world, which is something that we already discussed, why I disagree with it, which is what today happens. The supernatural is a priori excluded as a possible explanation of anything.
I've been listening to you yesterday, so I understand what you mean. So I will give you the answer that scientists usually give to this question. Okay. And the question is, what I need to see and how I can be convinced that intelligent designer exists.
Yeah, I asked you actually in our last
answer of this question is, as long as I am able to explain the phenomenon by natural forces that I know of, meaning chemistry, meaning physical chemistry, interactions. If I am able to explain the phenomenon by natural forces, I do not need to have explanation of intelligent design. This is the answer. And what does it mean, explanation, in that case? The explanation mean could be, even if I'm not able to perform the experiment to prove it, could be possible explanation or plausible explanation which is completely feasible to happen. If that is the case, therefore, I claim that I cannot need to have intelligent designer, or you can push the intelligent designer behind, completely behind in that case. My point is, as long as I'm able to explain it, even theoretically, even hypothetically, but what does it mean, explanation? So if I'm able to explain it, I do not need intelligent designer. That's it. So what does it mean, explanation, in that case? The explanation means something that contains completely normal, natural forces, which we people, scientists knows, explained. And also there must be explanation of the observations. If we see observations, you explain it, these observations, even that is hypothetical, it's still, you do not need intelligent designer to exist.
So when do you need intelligent design? When do you need intelligent design?
I explained to you. If I'm not able to explain something in any way, any hypothesis cannot explain natural phenomenon, then I can think about intelligent designer. Yes, so far there's no.
You can make shit up and then say it's plausible. So
no, it's possible. So far, there is no even one phenomenon, that I cannot potentially explain with normal, scientific, natural forces.
I can give an example of what you cannot explain by natural normal forces.
Excuse me?
I can give you right away an example of a subject which is very relevant in our discussion, which I actually addressed in my video, which I published yesterday.
If I'm able to explain,
it.
If I'm able to,
in a plausible way, in a reasonable way.
Yes. So if I'm able to explain something in reasonable way, forget about ID in this particular case.
And if you cannot?
If I cannot, theoretical, then I can say, okay, might be intelligent designer. But again, there is no such a case.
How do you know that?
Because I've studied biology all of my life. I studied chemistry, although I'm not chemist now. I'm mostly biologist. And even I'm specified, all the forces of nature that I know of are completely capable to explain origin of life, evolution, and today's world. Do you understand that?
Yes, I understand your position, Dmiter. And the question mark
the fact that you don't understand, and you have gaps in your knowledge, it doesn't mean that it's intelligent designer involved.
My position is not based on gaps of knowledge. My position is based on what we do know and based on the best explanation at hand in regards of the phenomena which we observe in nature.
I agree. The best explanation is the natural explanation. If I'm able to find natural forces for something, that's good enough for science to say there is no intelligent designer involved. That's it.
Okay.
So
I disagree with your position. I think that everything that has to do with abiogenesis is not in a reasonable manner explained by natural forces.
Why?
For example, I think that is an issue which I brought up already, and I don't think that the assignment of the 64 codons to the 20 amino acids. Assignments are always made by intelligence.
Because
that's what we observe. We know that intelligence is able
this is your assumption. Let me explain how the actual codons formed. Entirely natural way.
It's not the issue how codons physically form the triplets. The question is how has I will give you an example and an analogy. When you see a translation book of English to Chinese, then you have the assignment of the meaning chair to an object where you sit down. And the name chair in English is assigned in Chinese to Qizi. So we as intelligent beings, we can make that assignment of meaning. And the symbol, the code chair, making the translation to the word in Chinese. This is a process which is entirely performed by intelligent minds.
And you cannot explain that process. For example, the codon, or I understand your analogy for the translation.
Okay.
But what you want to say that you cannot explain formation of codon by natural forces. That's what you're trying to say.
The assignment.
Exactly. The assignment. Did you read
the amino acid alphabet?
Okay. Did you read Kunev's paper for origins assignment?
Kunin openly admits that this is a big enigma which science cannot explain. So
what is the forces?
You have there precisely the situation where a first-rated scientist like Kunin openly admits after his science paper and investigation that the origin of the genetic code is one of the biggest enigmas in biology.
Okay.
It has not been solved in the past 50 years, and that it will not be solved in the next forthcoming 50 years.
Okay.
So this is a very strong and important admission from a first-hand scientist, which is an expert in that field. So how do you deal with that?
Okay, let me explain how I'm thinking. Kunin and his review about origin of codons. Actually, he has 2 reviews of origins of codons. One, it was 2007, I believe. Another was 2017, probably 10 years later. Maybe the last one was 2017. Yes, it was, I think, was 2017, Kunin's paper. And he always talking about the big problems in the origins of codons. For example, he always may say about paragraph 22, catch 22 in the codon.
Yes. Yeah.
And those kinds of things. So I read a lot about that. But see, the position of scientists like Kunin, he's a good scientist, although he's completely wrong in his RNA world idea, but he's still a good scientist. The position when scientists discuss those things in the papers, they discuss it because there is something to be discovered. There is a gaps for discovery. That doesn't mean he said, oh, there must be intelligent designer immediately. What you're doing, you're taking papers of scientists, and because the scientists always point there is a problem somewhere and there is something to be discovered, you just catch this phrase that there is a problem, and you expose this phrase that there is a problem for discovery, and because of this, you start to make a big claim that it's impossible to be discovered. There is a huge difference between saying that, yes, there is a problem in science, how that happened, between that and to say, oh, there is no way to discover, there is no way to figure it out, only ID, intelligent design, must be involved. This is a huge difference. Neither Kunin is intelligent designer, neither any scientist that I ever know and I ever read. I read probably a few hundred papers for origin of life and also for genetic code formation. No one ever says
that's
that
precisely the problem which I point out. That's because the entire modern philosophical framework is based on methodological naturalism, where an intelligent designer cannot even be mentioned. And if you do so, then you can be sure that your peer review will not pass.
Well, that's completely wrong. You're completely wrong.
That's what we have. Do you know Mr. Sternberg? Do you know Mr. Sternberg? He's a very important scientist which had submitted a science paper from Stephen Meyer, and that paper was actually peer-reviewed by him and published, and he had serious problems because of that, and that is not a single case. So that what I am saying is not simply invented by me. This is what we have seen happening. So that is the problem why you will not find any science paper which will point to intelligent design as the best explanation in regards of any relevant question of origins, and not because intelligent design doesn't have a better explanation.
Why should have a better explanation since I'm able to find a natural process for that?
How do you know that you will find a good natural explanation?
Because I am able to explain step by step, although with not, okay, I can explain feasibility, how it's feasible, how it's possible, a process. I can explain the driving forces for that, step by step, entirely in natural way. How you telling me that because I am able to explain naturally everything, there must be intelligent designer?
Well, you can make an easy statistical calculation about the origin of the genetic code and the triplets and the codons, and then you will have an alternative explanation which is entirely based on random statistics. And the odds to have a genetic code, they are in the trillions and trillions and trillions and trillions. It's such a huge number. You don't understand the size of that number, and I do neither. So if you want to stick to that explanation, fine. But I mean, Kunin, he admits that this is not a reasonable explanation, and I think it is totally unplausible.
He admits that it's okay. So by what model? So you're talking about how it's possible such a complicated code to exist and to happen to be.
Why should there be
since it's so complicated, and you're talking with person that actually wrote down a paper how the origin of codon formation happened.
It's not
Do you understand what you're talking about?
The formation, the physical formation of codons, which is actually made by 3 nucleotides which work together to assign to an amino acid. The physical aspect is one thing, but the software aspect, that's an entirely different thing. We are talking here about assignment, which is not physical. The question is, how has the assignment, the meaning of 3 codons to a particular amino acids, been established without intelligence? That's the question.
It is question, but you don't need intelligent designer to have that.
How do you know that?
Completely, because I know what I'm talking about.
Okay,
I can say it step by step. The codon formation. So let's go first before coming to the codon formation, because your claims are completely wrong in several cases. So completely wrong is the statement that when you have a chemical system, so let me just try to refer. So when you have chemicals, everything degrades. Everything degrades, and there is no urge of any molecular to create a life. Is that what you said?
Yes.
So do you know that this is a very old question? It's called the question about the second law of thermodynamics. The second law of thermodynamics, and the controversial of the living system, which actually upgrade itself and maintain themselves. And at the same time, the second law of thermodynamics says that everything should go from low entropy to high entropy. So from order to chaos. And this is a huge problem. But you know what? That's a very old question. Even Schrödinger was positioned that question. And so the guy from the Discovery Institute, James Storr, tried to bring the same problem over and over again, even in the last video. And he's completely wrong. What is wrong in this? First of all, any living organism does not disobey second law of thermodynamics. They are constantly under destruction forces. I know very well because I'm coming from DNA repair field. I studied DNA repair, and you have let me finish, please.
Yeah, but you're moving from abiogenesis to evolution. You need to explain actually the origin.
Let me finish, please. This is very old question, and always was this question. Even Schrödinger, in his famous book about what is life, he actually pointed out very clearly that this is a very strange phenomena, how it's possible life to resist the second law of thermodynamics.
Correct.
And nowadays, back in 2020, the answer is very clear. It's called Darwinian evolution. This is the way how the organisms resist the second law of thermodynamics. And this is the reason why something goes from chaos to actually order, because of the existence of Darwinian evolution. And this is your answer. For some reason, you don't want to say this. There is a Darwinian evolution. Do you understand what Darwinian evolution is?
You're just making a claim, Dmiter. You need to back it up. And first of all, you need to explain how from basic chemicals and elements on a prebiotic Earth, there became a situation of disequilibria of cells, which actually need to be in a very disequilibria form, and rather to go to disseminate energy holds that disequilibrium, and that is a big problem.
No, it's not a big problem.
How do you get a proton gradient to drive ATP synthase, which is necessary to make ATP?
Okay, look at the situation. You always put the claim that something is a big problem. It's never been possible to happen. No, it's possible to happen. It is completely well understandable. And moleculars could serve even short peptide, or the short nucleic acid, even that are random, they can always, with interaction with peptides, can create situation where you have remembering the previous stage and go in advance. This is called a mechanism of evolution, of Darwinian evolution. Because of the mechanism of Darwinian evolution, you overcome the situation of the destruction, of the degradation processes, because you remember always your previous stage. You have information.
Nucleotides do not remember their previous stage, and they do
yes,
not
they remember previous stage if these nucleotides are ordered in a proper way.
How do you get to that?
Because of the stability. And you always forget, in all of your claims, you always forget term stability. The more stable situation is the Darwinian evolution situation. It's more stable. It's a called dynamically stable process. It's a very well described in the literature. It's called dynamically stable process.
Science doesn't even know how to get nucleotides on a prebiotic earth.
You have nucleotides in prebiotic earth because it's described, possible description. You have clear possible description.
I've never seen such a description.
This is a completely wrong claim. It's a false claim. I'll show it.
I made two videos in regards of the origin of nucleotides.
Look, there is experiments. Just go and try to read John Sutherland, for example, and many others.
How many different environments does he need?
Oh, no, this is not the correct way of thinking. Is it possible to be actual, real, natural way of synthesis of nucleotide? Don't try to go there and to say, oh, this is not natural. They create all of these extra conditions, which is not possible. No, all of these conditions are possible. That's why I'm saying you can actually experimentally describe. The evidence is the experiment.
I've not seen it. I have not seen it. I made two videos on that subject. I have investigated Sutherland, and it's not possible. And I am fully in agreement with James Tour.
Just 1 second. I'll be back because I have to go because of the coffee.
Okay.
Mike, let me show you.
Are my drawings distracting you guys?
No. Just close a little bit of your view. I have to find here John Sutherland.
You got something to show? Screen?
Yeah, I want to show in the screen, but let me see, where was that?
It helps me to doodle while you guys talk.
Yeah, so I
keep me focused.
So let's see. John Sutherland.
I can read it here. In 2017, English chemist John Sutherland formed nucleotides amongst all of the basic building blocks, lipids for compartments, amino acids for metabolism, starting with cyanide as a common initial substrate. However, this ended up requiring 6 separate bonds with their own unique geochemical conditions, and whose products then needed to be mixed together in a specific sequence.
Are you finding this, Dmiter? Oh, yeah, there you are. Okay.
I think I put it there.
In the science paper of him, The Origin of Life Out of the Blue, published in 2017. And then he goes on, even this degree of complexity did not supply required products, but only precursors. The requirement of so many unique bonds and associated chemistries in such close proximity to each other, of course, stretches plausibility.
Oh, Dmiter left for a second. Hang on a second. I'm looking that up. So if somebody had a link, I got a link. Okay, is Wiley a good one? Wiley be a good one? No, Wiley not good. Okay, origin life out of the blue, research. Download citation, requestful. Okay, when did this come out? It's 2017?
2017, correct.
Let's see. Oh, here's a PDF. I got a PDF link. PDF. Oh, maybe. I am not so certain. Nope, not working. Trying to find a link for that.
I'll give you a good link, Mike. You can put this here. And
make sure it isn't that bad link. Let's see. Okay.
Okay, so this is one older paper of John Sutherland, and this paper was one of many papers. He actually showed experimentally. I'm just trying to go back to
That's a different one.
Yeah. So, and in this paper, why starting? Okay, that's the other one. So this is different. Okay, so I'm here. Okay, in this paper, he explained all the completely understood chemical processes to formation of nucleotides. You have very clear scientific process of possible prebiotic chemistry where you can produce nucleotides. I will stop this paper now and go for another paper, which is this one. Is it changing? Yeah, it's changing. Which is this one. And in this paper, there is additional description how from formamide you can have nucleotides, sugars, and many amino acids. And also, you see.
Okay, does the paper explain how the right-handed sugars were sorted out from the left-handed sugars? Because, as you know,
just let me finish. Okay, so this is a next step. So John Sutherland shows how to make a nucleotides prebiotically. It's feasible. It's completely natural. And here you can see how can polymerize. Actually, in some conditions, you can polymerization process and make a very short oligo. So I'm stop sharing right now to say if this is chemical reactions possible in general.
Well, I made a very specific
I asked question. Is it possible these chemical reactions to happen?
Well, first of all, I didn't read the paper which you just linked to see what they are explaining, and okay. That's why I'm asking you.
Provided experiment.
Since you read the paper, since you read the paper, I would like to ask a very specific question, which is a problem of the synthesis prebiotically of nucleotides. That is, as you know, ribose is homochiral, and life only uses right-handed ribose. How have prebiotic mechanisms sorted out only right-handed ribose?
It's explainable, natural. It's called Darwinian evolution.
There was no Darwinian evolution.
Bottleneck process. It's called bottleneck selection. If you have one reaction, if you have one reaction, which it's happened to be, for example, with the peptide, which fit with the specific peptide, and only one form fits to this particular peptide, only one form, L or D form, fit to this peptide, This is called selection, bottleneck selection, meaning for the next generation, only left or right will be selected for the purpose. So you have complete...
There was no purpose at that stage.
Purpose is called survival.
There was no life to survive at that stage. There was just chemicals.
Life to survive. I explained that the life at the beginning is only 2 amino acids and few nucleotides, equal to life. The only reason that this particular system can be called life is because possess ability of Darwinian evolution.
Dmiter, you need to start right from the beginning. And I am asking about how right-handed ribose has been sorted out from left-handed ribose.
At the beginning, you don't have specific selection for left or right. At the beginning, after prebiotic synthesis, you have numerous 50 or 60 different types of amino acids, alpha, beta, gamma. You have all kinds of possible fatty acids. You have cyanide. You have all kinds of prebiotic species and molecules, left and right. If you perform just chemical reactions, you have equally left and right. Amino acids, left and right.
Correct, yes.
Now, but that is the moment that you completely and continue not to understand for some reason. Once you have Darwinian evolution, you have a system which start to select what is needed to be more stable and more stable. So it's all about stability. If something is more stable, will survive longer. If something is not stable, will not survive longer. It will perish. That's simple as that. And now, why you have only left amino acids? It's simple, because left amino acids fit to specific reaction. There is why you have a bottleneck selection.
And right amino acids also fit to other specific reactions.
Yes,
so you have
you
have a chaotic mix of different chemical reactions, but you cannot simply have them 1.5.
No, this is not argument. You have a completely feasible argument. It's a called process where you have a specific important, very important process. Let me assume that you have a very short peptide from left and right amino acids. Let me assume that you have initially Darwinian evolution just set up in very simple way as I described, just by interactions from peptide and RNA. And let's assume that you have both left and right. But at some point, this stable system will require only left to continue with the RNA replication, for example. Now you have next step. So you already have Darwinian evolution, and because of this, you have next step of selection. Now, that doesn't mean at the same time there was not adjacent or simultaneously existence of another systems that also was selected at first. But see, the process goes and some just falls down and another continue because of the selection process. So the answer of this argument that it's about second law of thermodynamics and all these arguments that there is no process that can go and create life, it's wrong. The process is called Darwinian evolution. The Darwinian evolution is the only system, the only process that can go further, can complicate and can advance.
Can you show me any kind of scientific paper related to abiogenesis which proposes Darwinian evolution as the driving mechanism for complexification?
All of them.
Do you have can you give me one where I can read about
first of all,
evolutionary Darwinian evolution as the mechanism? Can you show me the paper? Do you have an example?
See, everybody is talking about Darwinian evolution when you're talking in in origin of life. However, the problem here, it's not about is there papers or not about that. The problem is that there is a logical explanation. And for example, my paper in 2018, the first paper, it's all about Darwinian evolution. It's actually, this is the only idea. According to, for example, RNA world hypothesis, the Darwinian evolution became with self-replication ribozyme.
Dmiter, let me just to answer to Leophilius. Leo, who brought up in this discussion thermodynamics was Dmiter, it wasn't me, okay? Go ahead, please.
Okay, so it's answerable. It's possible to be answered. There is a feasibility, there is a hypothetical, although not proven directly by, I do not have ability right now, experimental ability. I don't have laboratory to go and show you directly how it happened. But that doesn't mean there is no possible explanation. It is possible explanation. It's completely feasible explanation. That's why you have situation. That's why I'm arguing. And the third paper that I provided to everyone here is about formation of Darwinian evolution. If you don't have Darwinian evolution, forget about life. There is no possible life. You said many times the amino acids. So let's go to the just codon formation. So
yes, that was our first investigation.
There is no reason
assignment of the codon triplets alphabet to amino acids. That was actually our first question, which I wanted an explanation, and we already moved to many different topics. And it would be great if we would just stay to that first questions which I asked. How do you assign the triplet codon alphabet of the genetic code to amino acids? How did that happen?
Okay, so again, I'm repeating your question, your side, to be in the same page here. So you said that there is no reason that assignment amino acid should assign for the specific codon.
I know only intelligence that is able to give an assignment, a meaning of something to something else.
Well, you're wrong, and 100% wrong. The reason that you have codon formation is because by codon formation you have more stable system. That's it. If you put the right.
Dmiter. There was no system yet. There was no system. What system was there?
It's a possible system that I described in my second paper, which is very clear. Once you put the correct amino acid in the correct position, you have more stable form of the system.
What kind of position and what kind of system was there at that stage?
Okay. At the moment you behave that I never discussed with you any models. I explain. I'm not going to explain the same model over and over again because it's not neat to do that. So if I'm able to just briefly to share for a few seconds. Yeah, this one. So here, Mike, can you accept my share? Okay. So here in this system, you have a situation where a specific amino acid is assigned to GC-rich RNA. This is already pre-codon formation. This system particularly creates a situation where you have a specific amino acid like arginine together with aspargine coming in the same place. Why? Because you have completely natural process of interactions between RNA and GC-rich RNA and free arginine in that case.
So they are interacting physically and chemically together, is that correct?
Yes, it is physical interaction because you have...
You do not have a physical interaction in the tRNA where the...
No, you need. This is analog. I explained hundreds times again, and you behave that you never seen that. I explained this is a model of formation of amino acid synthetase. Amino acid synthetase actually recognize the amino acid and put in the right tRNA, all physical interactions. Whatever you see as the model is a completely protoform, simple protoform, so simple.
So how do you go from that protoform where you have a direct interaction of the RNA with the amino acid to the situation where we have the tRNA, which is the intermediate between the triplet codon and the amino acid? And in special, you need to approach a very complex process, which I pointed out in our last discussion, to make tRNAs. That is a multi-step, highly complex and very specific process to have these tRNAs. And why would prebiotic molecular mechanisms come up with that biosynthesis process to make tRNAs if they have no function by their own?
That's not true. You have completely wrong claim here and statement. And I'll explain why. If you have the system which I described just before a few seconds, you have a situation where bridge peptide will be repeat and will come with higher frequency every time when it's coming. However, you have also a process of mutations that is happening all the time. And the best fit will select. Something that is more advanced will be selected. tRNA, okay, there is a very good model for the evolution of tRNA, and not one tRNA. The tRNA also can come initially.
Okay. So let me ask you, Dmiter. Let's suppose there was an evolutionary trajectory of the make of tRNAs prebiotically. Okay. So
no,
you have
there's no such a thing. tRNA, it's not made prebiotically. It's simply not possible. Your assumption is wrong. There is not possible to have tRNA prebiotically.
No, I made a supposition, Dmiter. I made a supposition. I want to understand why prebiotically or biologically or whatever, natural processes would form a biosynthesis pathway, a fabrication of tRNAs, if they at the end only have function in the completion of the entire system of translation fully developed.
I'm not quite sure. Do I understand very well your question? Can you try to repeat this question?
I think that my video, which I put right at the beginning in regards of photosynthesis, illustrated in a very clear manner what I am meaning, but I will try to put it in words. If you have a system of 5 parts, and all these 5 parts are essential and necessary in order to have, at the end, a product which is functional. Let's suppose in the photovoltaic system you have the solar panels, then the batteries, and so forth. You have 5 parts which you need, which are absolutely required in order for the capturing of photons through the solar panels, and in the end you have the battery to store the energy. So if one of those 5 parts is missing, you do not have a pathway which will in the end provide any kind of or sort of function. Now, each of these 5 parts also requires very complex processes and manufacturing processes to make each single part. Now, if you do not know the function beforehand of the last outcome of what you want to get, then making a solar panel by its own would be completely meaningless. So you would make an entire factory to make a solar panel, But the solar panel by itself, without the other parts, the battery and so forth, would have no function. And this is an analogy to what we see, for example, in regards of translation in biology. In order for translation to work, you need the ribosome, amino acid tRNA synthetases, tRNAs, you need messenger RNA, you need the genetic code for the assignment, you need all these parts. If one of those parts is missing, you have no translation, you have no function. But that is the crux of my point. In order to make the ribosome, you need complex machinery. You need over 200 assembly proteins, scaffold proteins, 75 cofactors in order to make the tRNA. You need complex machinery. Now, each of these parts individually without the other parts have no function. If you have only the ribosome and you have the entire manufacturing biosynthesis process to make the ribosome, but you do not have tRNAs or all the other parts, the ribosome by itself has no function. So why would biologically or by biological or prebiotic conditions, some biochemical process produce that manufacturing step to make a ribosome which would be non-functional without the other parts. And that is what I say as integrated complexity or as irreducible complexity or as interdependent complexity, where you have only function when you have all the parts working together in a joint venture. And if that is not complete, you have no function. And that is the problem.
Okay, so did you finish?
Yes.
Okay, so now just listen. First of all, your model is wrong. You are working with assumption model, which is wrong. We evolutionary biologists does not work in this particular way. By saying something is irreducible complex, it doesn't mean that you are not right. Yes, it's many things in the today's, if you see, they are reducible complex. But when we say something is reducible complex, irreducible complex, that means in the context of the system that something exists, meaning you may have something, some component of this complicated system, which is absolutely vital. And if you remove it, everything will collapse.
Yes, that's exactly what I'm saying.
At the same way, you may have another component, which when you remove it, the system will still survive. Now, listen carefully. When we say that something is reducibly complex, meaning that it's reducibly complex for the system that exists. Therefore, in more simple systems, you may have the same thing which is completely feasible to be independent from the other. Let me give you an example. I'll share you a screen now. So let's see what I have here.
So let me ask you, Dmiter, translation as is. What is the smallest system which is possible to provide translation as we have it today?
The answer is, just give me 1 second to share it. Share screen.
Well, just to
the answer is
just to ask my friend which is making a question. Excuse me, Dmiter. Well, in the first place, science has not provided any explanation whatsoever how life could come from non-life in the first place.
Please do not say that, because you're wrong. When you say whatsoever, there is no explanation. In fact, there is at least 100 different types of hypotheses for origin of life.
And none work, because if there was one then all the others would just be buried. And we would have just one explanation which works, but none of those which you mentioned comes even close to explain how life could have come from non-life. These are hypotheses which have not been demonstrated to actually work.
Did I say just before a few seconds, if you have natural possible explanation, let me see what happened here, just 1 second. I'm not quite sure.
Oh no.
Okay, I'll stop sharing because, okay, I'll share it again. So, did I say at the beginning of our lecture that if you have possible natural explanation, that is good enough for the science?
Yes, but you have not given so far any plausible narrative of a possible explanation.
No, I gave it to you. Okay. I gave it to you, and
so,
I explained
we will just agree to disagree, because I do not think that you gave a plausible
I explained to you at the moment, once you have Darwinian evolution, you can explain the next steps. It doesn't matter that I don't know exactly by nucleotide how happen these steps, but you can explain every single step. And let me finish with my presentation for your irreducible complexity.
Okay, go ahead.
Because you are completely wrong here. And don't use a cliche, because you're constantly using a cliche like this is never, it's not possible to be explained. There's no even possible at all. This is a cliche from the Discovery Institute. Please stop listening this guy. Because
this is not true. As I told you previously, most of my information is not coming from the Discovery Institute. So
yeah,
please assign or attribute things that which I didn't confirm or say.
Mike, I'm trying to share something here.
Oh, I'm sorry.
Here we go.
I just switched our the icon for this video to that drawing.
So let's see. This is a play cards. You see the play cards? Okay. And as you see, the play cards representation, the entire pyramid that you can see here.
Okay.
It's the representation of any living organism. Let me say this living organism is a very complicated organism.
Okay.
Each card, each card represents some sort of, let me say, protein or process. Right?
Okay. Okay.
Now you have pyramid where there is some processes or cards here which are on the bottom.
Okay.
And every card on the above depends on this from the bottom. Is I'm correct?
Okay.
So you want to say something like that. If you remove a card from the bottom, pull it out from the bottom, the entire system will collapse.
Correct, yes.
Yes, and that's what you want to say about your irreducible complexity.
So do you think with that illustration you have actually refuted my claim?
Are you agree with me or not?
No, I am asking you, with that illustration, how are you refuting my claim of irreducible complexity
yes, so according to your model, which is correct in that case, if you remove a card from the bottom, the first row on the bottom, everything will collapse.
Right. Okay.
So that's your model. You just said that's irreducibly complex. You cannot explain if you remove just one part of this super complicated system, everything will collapse.
I would like to make another exemplification. Okay, if you have an entire car, but just the piston in the engine is missing, can you use the car for the purpose for which it was made?
Yes, you can do it. Just listen to my example first, because this is a simple explanation of what you want to say.
Okay.
Now, you see this guy pulls from 1 to the 3rd row of a card, right? So if he pulls, let me assume there is no vibration. Just eliminate for the heck of the thought experiment. There is no vibration, meaning if you pull the card, you will not incidentally by your hand will demolish the entire pyramid. So if you pull the card from the 3rd row, what will happen?
It will collapse.
So all of the above 3rd row will collapse, and some of the left also will collapse, but there will be some small section which is under the card that will survive, that still will have a pyramid. Is that correct or not?
Yes.
Okay. Now, by saying irreducibly complex, so I'm stopping sharing this at the moment. So this pyramid is your example that you're trying constantly to come with. And please, this example for irreducible complexity, it's not something that can help you in order to provide against the evolution. Because when you pull the card from the bottom, everything will collapse. That means a process of protein, which is absolutely vital and important, if you remove it from the organism, it will die.
Correct.
However,
it will die. Correct.
Yes. However,
and you have a major set which is absolutely required to have life as we know it.
Let me say something like absolutely butchery way. If you remove the heart of someone, he will collapse immediately.
Yes, and now put
I'm sorry about this analogy.
No. Now you put a protein in a first living cell, which is absolutely essential. Which one would you agree, which is absolutely essential for life to self-replicate?
Let me finish. So you're going forwards too fast, and by going too fast, I cannot explain everything. Okay. So because you're bringing up the irreducible complexity all over the way, in every way. And I'm trying to show you that your model is wrong. Okay. We evolutionists does not work in this particular way. In fact, if you remove the card or remove a very vital process, the entire organism will collapse, and we agree with that. That's why you observe processes like mass extinctions, for example. The people do not survive in some specific environment. It just happened, changes in the environment, and they do not survive because something vital is blocked. It's eliminated. That's why the organisms die. However, if you pull out the card from the top, from the peak, from the summit of this pyramid, the pyramid still will be there.
Yes, but you need an analogy which is equivalent to what we see in life. And I think that the factory would be a much better analogy because we know by scientific literature that cells are factories in a literal sense.
I will come to biological analogy. Okay.
Yes, but you need an analogy which is very similar to what we see in life. And I think rather than putting a pyramid of cards, you should eventually provide as an example, as an analogy, a human-made factory, actually a self-replicating human factory.
Can I finish?
Yeah.
Okay. Let's finish with this analogy, because this analogy is important to put my perspective and point how we evolutionists actually working. So your model of irreducible complexity is here in this huge pyramid. And you can, according to you, if you pull from the bottom, everything will collapse. But also, if you pull something from the top, the very top one, it will collapse just the neighbors, but the entire pyramid might stay.
Okay, correct.
So as down you go through this pyramid, as dangerous for the pyramid will be if you pull something. The same thing happens in the very complicated living system. You have organism, which is very complicated, and you pull out something. Let me say you change a codon, which reconstitutes arginine for alanine, for example. It just changed this codon. And the organism will die. Why? Because too many proteins are already attached to this specific position to be arginine, not alanine, in order to maintain all of the biochemical processes. So the actual codon from the bottom, it's so important that everything above will collapse if you remove it. And everything, as I said, up to now, is according to your model of irreducible complexity. However, when we say that something is reducibly complex, we meaning in a more simple system. What we mean that the same card that you pull from the third rod, for example, if you pull it out from the third row, just imagine that your pyramid is only 3 rows, not 6, not 7, just 3 rows. Now, if you pull the same card, identical card from the third row, how the same as this guy did pull it out, now you have a system that will still survive. So this is how we evolutionary biology working. So it's very important to understand that we using reducible complexity not in a steady state situation, but in an evolutionary situation where you have a more simple system and more complicated system. So to say that something is very complicated, it doesn't mean it's reducibly complex. In more simple forms, could be very simple and could be still functioning. For example, who said that some chemical component cannot emit a specific electron and just harbor one particular peptide and just charge?
Well, I would like to answer to your analogy which you made with the cards. Can I? Before you start with another topic. Well, first you need to have the pyramid by natural means. I think with that observation, your entire analogy falls down.
Observations are everywhere. Now, if you
see... I am asking, how do you get your pyramid by unguided natural means? Because we know that people, they can make that sort of pyramid which you showed the picture, but we do not know of any natural mechanism which would be able to make that system of that kind of pyramid.
Is it cyanobacteria? It's more simple than Zea mice. I mean, then, for example, high plants.
No, the threshold.
Do you think that, for example,
the threshold is not cyanobacteria. The threshold, which is to go from, in my understanding, I mean, for you, even an amino acid or so, you think that's already life. For me, I go with the explanation which Davis gave. For me, the threshold of life is what we can observe is the smallest living cell which is actually alive today, which we know is, for example, Pelagibacter oblique, which is a prokaryote with about 1,300 nucleotides and 1,300 proteins. More or less, that kind of complexity. That would be the complexity which I call irreducible. Why? Because if you remove just one of that proteome set, you cannot have self-replication and life as we know it. So my question is, as an analogy, Dmiter, if we take a car and you have a car which has all the functionalities to perform what it is made for, to work with gasoline and so forth. in order to work, you need a minimal number of parts that if you remove just one part, you do not have anymore the functionality which it has. So if you remove an essential part like the piston of the car, I just want a yes or no from you. If you remove the piston of that car, do you still have the functionality it was made for, or just will it stand in the garage and you cannot even remove it from the garage?
I can tell you something very simple. You are correct. If you remove the piston of the car will no longer.
So, yes.
But,
no, Dmiter, I want to answer to that now. I want you agreed that if you remove a piston from a car, you do not have anymore the functionality of the car. Now, I think we agree that you cannot make a car without intelligence. You have to project it. You have to make all the integrated functionality of the single parts, bring them together in a functional, meaningful way to have the functionality of that car. Okay? So the same applies to a living cell. You have a minimal set, a minimal proteome, a minimal a metabolome, a minimal genome, which is absolutely required. You need information, matter, and energy to have a self-replicating living cell as we know it, that it works. Now, if you remove a helicase, just one protein, you will not have that system working. You will not have self-replication anymore. If you remove ATP synthase, you will not have production of energy anymore, and you will not drive life. If you do not have nucleotides, you will not have a genome, which is the hard disk to store information. You will not have life. These are just 3 examples. But we can go through all the 1,300 proteins in that minimal cell, and if you remove just one of these proteins, you will not have life. Now, this is the crux of the issue. How do you get, for example, just a helicase by itself? If the helicase by itself has no function, you would not have, you would not make helicase if it has no function. You would not make a piston.
I understand your point.
If it doesn't have function in the car. And
can I get a word?
Pointing out.
Could I get a word in, please? Okay. First of all, Immutable wanted you to comment on 2 comments that he put up, Dmiter and Otangelo. You got to quit thinking about car mechanics and factories when you're thinking about biology. Okay. We kind of have a rule on this channel: no more goddamn analogies. Is that fair? Let's talk biology.
Well, but when I make analogies,
I know when you make analogies, you're thinking badly.
I try to simplify what I want to communicate in a way that any one of us can actually understand it. And
yes, but we can simplify things to the point of losing the point. And that's what you're doing. You're thinking like an engineer. You're assuming intelligent design. You do it all the time. That's why you use analogies. You creationists always use analogies. Stop with the analogies. Okay, I got a rule on my channel. No analogies. Well, I use billiard balls, so I guess I violated my own rule.
Either we describe things as they are, or we don't. Either we try to exemplify things in order to understand what happens.
Okay, but a car with a piston is not a living thing. It's not a living system. It doesn't have ancestors.
Okay. Do you know the difference, Mike, between a human-made factory and a cell factory? Do you know what the principal difference is?
Yes, the cell is a hell of a lot smaller. It's a living system, and it works with molecules, and it has nothing to do with humans or design. It is not designed.
Okay, Mike. The main difference is that cells self-replicate while human-made factories do not.
But it's a little more than that. There's more than that. They don't just self-replicate. They self-replicate with modifications. Okay, they self-replicate in a system that filters out whether or not something has gone wrong with them. They are stochastic. They are they add up to a statistical curve where they either work or they don't, and when they don't work, they die. Okay, And that's the difference with living systems. We don't build cars that way. Cars do not, we don't put the ingredients for cars in a factory and wait for them to happen. Okay. We know what we want in the end. We know how the thing is going to fit together. We design them intelligently.
Mike, if you want to understand my point, then you guys need also to try to listen what I am saying.
Oh, we know we have listened. We have listened so much. We understand what you're saying so well.
Mike, in this now, I don't think so. In this whole conversation with Dmiter, we had 3 sessions basically. I have difficulty to bring my point because Dmiter is basically talking all the time, and that is difficult. And I think the point which I made right now is very relevant. And you say no analogies anymore, but I think the analogy which I made is very pertinent to what we see in life. So I don't know why you try to avoid that I make that kind of analogy, because
you're trying to make it. You're trying to make an argument by creating. You're trying to make an argument by using an analogy that we know was intelligently designed, and you're trying to prove intelligent design with an analogy, and it doesn't work. Anyway, could you comment on Immutable? Wanted you to comment on these two pieces of text.
Okay. So
I don't.
Which are reasonable for replicating DNA are probably at the bottom of the pyramid. Yes. Any metabolic processes feeding off autocatalysis would likely be placed at the middle of the pyramid. Yes. So at the very beginning, you have. So to answer this question, I can say at the beginning of life, when you have only Darwinian evolution with a few amino acids and just short RNA, it depends on the environment supply. The way going from this point, the very beginning, to the Luca is a way of independence of the environment, meaning once you have ability to produce and to make metabolically some specific component like another amino acid or ATP, for example, you became independent from the actual environment. But I want to go back to the Otangelo's car, because this is he want to use this particular example, and I can tell you how the evolutionary biologist thinks, how they consider the analogy of the car. So let me assume, Otangelo, that your car is without piston. As you mentioned, the car will stop working.
Correct.
As a car, right? We agree with that. Is anyone mechanic that will disagree?
Okay.
No, nobody will disagree. Correct. And now I'll tell you how we evolutionary biologists thinks about it. This car, it's happened to be in a field where there is a farmers, and in these farmers, actually, this car became absolutely fabulous chicken coop. In this car, it's happened that this particular farmer became growing chickens and actually lay absolutely fabulous and very good legs on the seats of the car. Here is the new function of the car, the absolutely new function of the car, and that's how the evolutionary biologist thinks about it. You don't have a system which is already set up to do everything all over and over again in the same conditions, and once you change and you mutate it, it might happen a situation where this system to be in environment more suitable for another function. In that case, the car will no longer be a car, but will be absolutely fabulous chicken coop. So I will grow up my chickens in this car in the field, and I will be happy farmer. What about that? So see, it's not about you have just that function. It's all about what will survive, what will happen next step. The same thing happened with the Douglas X. He trying to say, if you remove a specific, so he was trying to study in Harvard, when you remove a huge chunk of DNA from the gene which is responsible for protein of the wall, of the bacterial wall, and this gene is responsible for resistance of this particular bacteria. He said, look, I put the bacteria, and now this bacteria can no longer make information. It's no recovery of the information. Of course, will not. We are doing that all over again, every day in the science. The problem is, and how we evolutionary biologists think, if you take this deleted bacteria with this deleted chunk, it might happen much better suitable if this bacteria stay in another environment. The same thing with the car. It will be absolutely useless as a car, but will be wonderful, wonderful for purpose of chicken coop. That's it. That's how we evolutionary biologists think. And that's why the pyramid that I'm trying to explain to you, the pyramid analogy, was that you always have a pyramid which might have less layers actually that you can have from this particular pyramid. So if you have more simple system, of course, back in time, there was, so as I said, the genetic code, of course, determined the protein sequences. But back in time, there was no even proteins. There was just a short peptides. Therefore, you don't need that complicated genetic code. In fact, if you see the genetic code, you will see that the genetic code is predominantly designed in designed. Did you see designed? I'm saying it's designed by 2 nucleotides in the codon. The third one is wobble and doesn't matter too much. It matters, of course, but when it matters because of another factor which is called EFTU. EFTU is a factor in the ribosomology. If you study ribosomology, EFTU dramatically increased the recognition of this particular third nucleotide of the codon, which was wobble. Now, the amino acids, it's mostly determined by the first two nucleotides, and you can see that in many papers. Therefore, the system can function in a much simple code with 2 nucleotides. In that case, you don't need all the amino acids because you don't have that complexity. Let me say what is important for example, in the system which is so simple, like whatever I described, one peptide, and one RNA, which is very short. What is important for this system is just to be more stable, nothing else. It's more stable, that's it. That's why if it's happened to be that particular amino acid, it's in this position, it will interact.
In my understanding, what would happen, that system would just lay around on prebiotic earth and nothing else would happen.
Okay, I'll explain you.
In the same sense that if you would have, in my analogy, just a piece of metal laying around on the floor, it would just be there and then become rust. It would not by itself complexify to become a part of a machine and then a machine and then a factory.
Okay, don't forget. You don't have steady state Earth. You have heat, you have a cold, you have rain, you have wet, you have dry.
You would have just a random state of molecules laying around. And that's
exactly. You will have random state, but not if something is more stable. The combination between this particular amino acids and this particular RNA is just more stable in heat environment and wet environment. And
so by having just some parts in a stable form, does that somehow bring you to a machine by natural means?
It will bring you to functional system, in more complicated functional system.
Do you have an example how that happens? Do you have empirical evidence that that could occur?
What do you mean empirical evidence could occur?
I mean, I am now in a very difficult situation, Mike, which basically makes my continuing at your channel meaningless, because you do not want that I provide the way how I think. You want to basically prohibit me to bring forward the way I understand things in the biology, and I see no meaning why to keep here, because you guys are not willing to actually listen to what I have to say. So this is a monologue of Dmiter, which all the time tries to explain how he thinks things work. But if I am not able to explain my understanding of things, and the best way is really through an analogy, then I think my participation here is at an end.
Well, let's see. I think we've let you explain quite a bit here, but I'm not sure how much good it's doing.
So, Mike, I will be very honest with you. I think that we had several hours of interactions together. I have explained my entire understanding of biology in a very clear manner. I actually made even a video to give you an answer, and I think we have simply to agree to disagree, and there are simply two worlds which are not mutually exclusive, and of course one is true and the other is not true, but I think there is no way that we are coming closer to one actually agreeing with the other side.
No, so, I think your mind is completely closed to any kind of science, and there's no breaking it. I mean, I've kind of given up. I mean, I like you, Otangelo, and but I think at this point it's just a matter of us doing videos with basically responses to each other. I think I've understood everything. I've looked at your blogs. I've looked at your information. I understand more about how you think than you think. And I think I don't know, Dmiter, do you think that there's any percentage in this going any further?
I was trying to explain that by thinking in a wrong model, which it's not the model as evolutionary biology works.
Well, you start from the presupposition that the evolutionary model is correct, and I start from the and my whole interaction is not from a presupposition, but from a conclusion to which I came based on my understanding of how mechanics work, and I see very clearly a parallel of mechanics made by humans and mechanisms and mechanics in biology. Now, I think that the evolutionary model is not capable to bring up the complexity, the machines, the integrated complexity, the irreducible complexity, the interdependence of parts in biology through natural means. I respect you guys. I respect your views. I understand. As you understand my view, I understand your views. I believe that what you propose is not plausible. It is not possible. It is not compelling to me. But hey, that's how life is. Each one of us has a different understanding of reality, and I agree to keep interacting with each other, to be friends. You guys can come to my channel. We can keep having a dialogue. But at this point, at which I'm basically censored to bring forward.
Okay, I didn't mean to censor you too heavily on analogies.
So, Mike,
but, I don't know.
Otangelo, let me say why I think the evolutionary model is correct, because there is evidence to be correct. For example, if you try to compare the DNA.
If there is evidence to it, then why has science not at not being able to come up forward with even coming close to explain how life could have emerged. That is the problem. And you guys keep thinking, oh, but we have explanations which are plausible, and if not now, but in the future, there will be a natural explanation. And I think that is simply not plausible. Now, if you guys think it is plausible, as I said, I respect it. It is fine with me. I mean, each one of us has the right to think as you guys want. But whatever you said, Dmiter, in this whole interaction with each other, is that based on my understanding of mechanics, of how things work, of how things can be done, and how machines and factories can be constructed, the model of evolution, in my understanding, is not plausible. That is my conclusion. And you can talk now another 5 hours or another 10 hours. We can have more 3, 4, 5, or 6 sessions or whatever. And our mindset upon what we have interacted already together, I think it will not change. But as I said, I respect you guys. I respect your views. And it's fine with me. It's no problem. We can keep interacting with each other. But at this point, I see no meaning to keep this going. Very honestly. I think I made all the efforts.
Well, Otangelo, that's something we agree on.
I think I have brought forward all the possible ways to explain my point. You guys don't accept it. That's fine with me. I'm not here to try to convince you guys. I've been here to make my point and to leave it to you guys to accept it or not. That doesn't change our interaction. As I said before, we can keep the dialogue. But I think at that point, it makes no sense to keep that going.
I just want to ask you probably one last question. Why intelligent designer designed all living creatures with absolutely the same or similar genetic code?
Well, as you know, the mitochondrial code is different than the universal code. So there are about, I think, about over 30 different genetic codes.
Yeah, it's not the same. I agree. There is a lots of variants in the code. It's not identical.
Yes. So it's not the same. So what is your? And I mean, as you guys know, and which I pointed out in my video, that the universal genetic code is more robust than 1 million different codes which have been experimented with. And the universal genetic code is extremely robust. It's more robust than over 1 million alternative codes. How do you explain that by natural means? Were materials just selecting and trying, attempting until finding the almost perfect or more robust genetic code? That is something which I simply don't understand.
That type of question is something that occurs after life has evolved. The type of thing.
If you don't have a robustness, immutable destiny, then you have too many mutations and you have no life. And as you probably know, as biochemists, only the ribosome has 11 different error check and repair mechanisms. And if they are not in place,
yes, that's why.
Then the error rate will be far too high, and you will never have a functional protein.
So that is why. So that is why in the comment that I had Speed read out earlier, why I said that proteins that are responsible for the fidelity and replication of DNA are likely the ones that are first to form. So that you do have some stability.
First, you need the entire system to have proteins, and that's a huge problem. You know, there are over 25 different macromolecular systems involved in synthesizing proteins. And even if you have, after that trajectory, a functional protein, you still need to tag it. You still need to have mechanisms where the cell needs to know where it is employed in the cell. Then you need the transport systems to bring it at the place where it is employed, and all that kind of complexity you need to have right at the start.
So, Otangelo, this is a question I asked at the very beginning of the stream. and I'll ask it again, is that do you think that proteins have inherent chemical activity? That the chemical activity that proteins have is independent of the environment that they're placed in?
Well, first of all, a protein has no activity or function outside on its milieu inside of the cell, each one at the right place, operating in the same sense as a piece of a machine does not have a function outside.
So I will partially agree with you in the sense that proteins, based upon the structure of their active site and based on the composition of their helices and beta sheets, will tend to associate based upon properties of charge and based on properties like hydrophobicity and hydrophilicity. So proteins that are more hydrophobic will tend to be ones that are associated more like near a cell wall or something like that. But it might also be the case that some proteins have a very strong affinity, that is, their active site has a very strong affinity for some metal, like iron or calcium. The point of this being is that, like you said, proteins have chemical affinity based upon where they are in a cell, but also based upon what they are equipped to handle. So the purpose of this being is that autocatalytic cycles, which are cycles that keep repeating themselves in terms of forming product and reagent, are going to be susceptible towards proteins and compartmentalized by them. Because proteins that have an affinity towards that type of reaction are going to associate and catalyze them even further and ensure that the fidelity of those reactions keeps going.
You are making huge assumptions here.
No, all I have made the assumption on is that, number one, proteins have a catalytic activity based on the type of molecule that they are able to catalyze a reaction with.
How do you
that is the
there?
first.
How do you get there?
That is basic biochemistry, Otangelo.
No. What I am asking, what I am asking is, I give you an example. Let's suppose by the mechanisms which you propose, you get to an ATP synthase turbine. Okay. Now, what good would be ATP synthase turbine good for if you do not have an energy gradient in order to feed it with ADP?
So the function you're asking about is, why would you have ATP synthase if you don't have an energy gradient to drive that reaction?
Correct.
Well, let's consider this, right? The reason that energy gradient exists in the first place is to allow that reaction to happen quicker.
Oh, so
okay.
Wait a minute. So
I wasn't finished.
Did lipids have an intent to form an energy gradient? Would the intent in the future to have ATP synthase to make energy, to make ATP molecules?
No. The intent is to lower the energy. So the intention is to increase the disorder. Hold on. If we go back to the second law of thermodynamics, what it states is that the second law of thermodynamics acts on systems to create disorder. So the way you do that is that in chemical systems, when chemical systems become more complex, they increase the entropy of their environment. So that would encourage something to create or to distinguish an internal environment, like the internal of a cell, from something like a lipid or a micelle, right? Because now you have a system which can be contained, and you have an area where that energy can be dispersed.
There is no thermodynamic necessity. It's actually contrary to thermodynamics that you form naturally, by no reasons at all, an energy gradient and membranes which have a difference of energy content to the outside of the cell.
Yes, you do. Do you know why part of the Krebs cycle is energetically favorable?
Go ahead.
It's because within the Krebs cycle, you have species that when they react, it releases carbon dioxide as a product. And carbon dioxide being released is energetically favorable. That is increasing the entropy of a system.
Yeah, but what I want to know is, why would prebiotically form an energy gradient of membranes? Why?
Because it has to increase the entropy of the system.
Why? But there was no system yet.
Yes, there is. What you have is you have a system like a lipid system, and you have RNA and small peptides.
You need to start from scratch.
No, the thing we're discussing is already well explained. There's tons of research that explains that small molecules such as nucleotides and such as lipids are permeable to small molecules.
What?
I won't repeat what I just said.
What?
Small molecules.
Nucleotides are what? Nucleotides?
Are permeable to cell membranes.
Okay, there were no cell membranes yet. There was nothing yet. You need.
And what I asked you, and so what I asked you from earlier, in our earlier conversation, is do you think that lipid membranes form naturally? Because I can demonstrate that right now. I can demonstrate that in a lab. That's a process that a first grader can do, Otangelo.
Okay, I agree with you on that, but my question was...
But so let's go back. Hold on.
No, hold on. You're right now. How do you go from a vesicle, from a lipid vesicle, to an energy gradient?
Because you have some molecules which are hydrophobic that will span part of that membrane. When you have small peptides, for instance, that are hydrophobic being that they do not like being near water, what do you think they will do? Well, what they will begin to do is they will begin to find places that are hydrophobic. For instance, lipid layers.
Okay, go ahead.
Right. So that is how you begin to have things like lipids or peptides that are beginning to span a cell membrane. It is more energetically favorable for some of those proteins, or for some of these peptides, or for some of these peptides that are very hydrophobic to be part of that system than to not be part of that system.
Okay, let's suppose that you have an energy gradient. How do you go from there to an ATP synthase protein?
Well, like you said, you already granted the fact that when we have that system, that there is some mechanism in place, be it a small cyclic peptide or whatever, that catalyzes that reaction. Well, and so the thing is, right, there's one of two things that can happen. Either you can have subsequent addition to that protein, which stabilizes the core of it, so it prevents other things from intervening with that reaction. Or you can have compartmentalization, where based upon the properties of that system that's performing the chemical reaction, it will associate itself to some part of the cell or protocellular environment.
Well, I ask again, you know that ATP synthase is a turbine which has at least 5 individual parts which are absolutely essential in order to provide its function. And
yes,
individually, the single parts, they have no function. They only work in an integrated manner as a system in order to have ATPs and produce the molecules.
Right, but so let's ask the more fundamental question of, if I were to begin dissecting an ATP synthase molecule, what is the most fundamental part of that molecule that is responsible for the activity of it? And the answer to that question is, as you were postulating earlier, it is a small cyclic, it is a small part of that protein which is able to house the ADP, so the adenosine diphosphate, and it is able to accommodate the...
Remember as well, Immutable, that there was no ATP yet. You need to produce your ATPs. How do you do that? You need energy to make ATP, and you need ATP synthase to make ATP. This is a catch-22 problem.
Well, again, the energy is feeding off of subsequent chemical reactions. so chemical reactions that are autocatalytic, that energy can be used by other systems. Because the type of energy that we're discussing with chemical reactions is thermal energy.
So I made a very specific question.
Yeah, and I answered it.
I don't think you addressed my question. You need ATP synthase to make ATP, and you need ATP to make ATP synthase. So what came first?
So you need ADP, adenosine diphosphate, to make ATP, and you need phosphate. And the reaction that forms ATP, adenosine triphosphate, causes those two molecular species to be in close enough contact and to form a new bond.
And which molecule transforms ADP into ATP?
ADP.
No, ATP synthase.
No. ATP synthase forms because it has an affinity towards ADP and a phosphate group. That's why it's catalyzing that reaction.
Okay, you need ATP synthase to transform ADP into ATP, correct?
Let's go a little more basic. When I have the compounds ADP, so adenosine diphosphate, and a phosphate molecule.
Okay.
Essentially, what is the process doing to what is happening within that enzyme in the transformation of adenosine diphosphate? And the short answer to that is adenosine diphosphate is entering some portion of that molecule. It is stabilized there, and for very technical reasons, the environment is such that...
That's not what I'm asking.
Right.
That's not what I'm asking. I am asking how can you go from ADP to ATP without ATP synthase?
And I'm saying you don't. I'm saying what I'm saying is that adenosine triphosphate, ATP, forms because ADP exists and because small peptides can form that have an affinity towards ADP.
So you claim that some small peptides interact with ADP and somehow they add a phosphate and make ATP without ATP synthase. Is that correct?
So what I am claiming is that as long as you have ADP, and as long as you have some small as long as you have some small peptide which has an affinity towards ADP in a very particular part of that molecule, particularly being the phosphodiester bonds, then you will be able to form ATP. Because what is happening...
So in that case, you don't need ATP synthase. You don't need all that complex machinery. Why has ATP synthase even evolved or emerged if you can have it in that way which you just explained right now?
So you're thinking about it in a wrong way. And the reason I say that is ATP synthase didn't form just randomly, right? That protein, and this is the question I was asking earlier. Hold on. And like I've tried to explain, right? And then the question I asked earlier is crucial to this. And you agreed. Proteins have activity based upon what is available to them in their environment, right? So proteins that have no chemical activity are going to be degraded. But when you have ADP, adenosine diphosphate, you have some library, so you have some variety of chemical species that exist, and some of those molecules, as they're moving around, are going to recognize that ADP molecule and interact with it in some type of way. Okay. It could be that they are relaxing the phosphodiester bond. It could be that aromatic residues on the protein are stabilizing with the nitrogenous base. It could be any number of things. But the important aspect is that properties of that peptide inherent to it have some affinity towards ADP as a molecule. And because of that, some of those proteins which are engaging with ADP will have a tendency to stabilize the phosphodiester region of that molecule. And because of that, what will happen if a phosphorus molecule comes in is, now that the energy is free and available enough, it will allow ADP to be transformed into ATP.
Okay, so you have ATP, so what?
So now that you have ATP and a system that can form it, now you have more energy for the system to use. Because now that energy can be formed to make longer proteins, you have the ability to move proteins to other compartments of a cell. You have more free energy for a system to work with. It means that now that you have energy, the system is not entirely reliant upon the upon autocatalytic cycles.
In addition, I just want to mention, so you gave a very good explanation that just the small peptides could produce there's some noise somewhere, I don't know. What I'm trying to say here is that he explained very clearly that. Hello, Tangelo, are you here?
Yes, I'm here. I'm listening.
Okay, I'm sorry. So that even small peptide that can have affinity to ADP can produce ATP just because there is some small gradient through the existence membrane. Now you have to pay attention also from the evolutionary point of view. In order ATP to be functional and to have a greater role, you need to have already folded long peptides, which according to any scenario of evolution is a little bit just a step after the first peptide formation. So the first peptide formation even don't need to produce by its own ATP, because environment there are lots of already activated molecules. So it depends from the environment, but they are already activated by supply from the environment, and that needs just energy from environment. And the second, once the short peptides or a little bit longer peptides start to produce ATP from ADP, it's not necessary to be super efficient process. But as efficient became, as more efficient became, as more advantageous will be to the system, therefore you'll continue to have improving and adapting the system and also complicated the system, just because there is a necessity and just because became more efficient process. And basically every single chemical process, biochemical process in the cells, goes from less efficient to more efficient and more accurately performed. The same is true for everything, like replication, for example. The very first RNA replication process could occur just by small peptide that contains 2 magnesium ions.
Okay, I have a question to immutable. The simplest RNA protein is ribonuclease, and it has 124 amino acids and 19 of the 20 different ones that are considered essential for life. How do you get from these simple peptides to the first functional ribonuclease?
So the way that's happening is that some of those, so it really depends on the structure. But the short way of explaining it is that some amino acids within that protein structure are there to interact with the environment, so say water, and some are likely going to be there in order to interact with other.
How do you get the 19 ones used in that enzyme, that RNA enzyme? How do you select it? Because in the environment there are hundreds of different amino acid species which are not used in life. How do you make the first selection process to have the ones that are used in that enzyme?
Okay, so that's a 2-step process. So the first part is prebiotic, in that, number one, things that are happening outside of this lipid system, right? There's hundreds of thousands of amino acids, but they're not all in the same concentration as each other. There's some that's going to be in slightly higher concentrations, some that's going to be in slightly lower concentrations. And among that, there are going to be some chemical reactions that are causing others to degrade. So one of the things that's happening to help kind of cause it, to make this easier, is amino acids that are high concentration, so that are in an overwhelming abundance over others, are going to find their ways into cells more easier, simply because there's more of them. And the second part of this is that once the amino acids are in a cell and they are reactive or interacting in some kind of way, right? Then the second part of this becomes, okay, how easy is it for this amino acid to be regenerated? How easy is it for you to add an ammonia back to this molecule? How easy is it for this molecule to be reformed?
I think you are not considering what I will read to you now. Amino acids used for life have amino groups and carboxyl groups.
Yes.
To form a chain, it is necessary to have the reaction of bifunctional monomers, that is, molecules with two functional groups, so they combine with two others.
Yes.
If a unifunctional monomer with only one functional group reacts with the end of the chain can grow no further at this end. If only a small fraction let me finish. If only a small fraction of unifunctional molecules were present, long polymers could not form. But all prebiotic simulation experiments produce at least 3 times more unifunctional molecules than bifunctional molecules. So how do you overcome this to get a minimal ribonuclease with 124 amino acids?
So, like I was saying earlier, right? And I'll try and explain it again. In the environment outside of a cell, you have hundreds, if not thousands, of different amino acids. Some of them have one carbon between the carboxyl group and the amino group. Some have 2, some have 3, some have 4. There are plenty of them. And
so how do you select the ones that are bifunctional, which can be added to the polymerization?
Good. So the simple answer to that, again, is number one, if you have, so for instance, let me take 2 examples. So I'll call these 2 molecules alpha alanine and beta alanine. Alpha alanine is a molecule that differs from beta alanine only in one significant area, and that is there is one carbon separating the carboxyl group and the amino group between an alpha alanine, and there are 2 carbons which separate their carboxyl group and the amino group from in terms of beta alanine. So one of the things that can determine selection is 2 particular factors. Number one, if alpha alanine, so the one that only has 1, is in higher concentration than beta, alpha alanine, by virtue of just existing and being bumped around by other molecules, is more likely to find itself inside of a cell membrane than beta alanine, because there's less beta alanine than there is alpha alanine. And the other thing is that with amino acids, you have what's called rotation about a carbon bond. And that's important because this rotation between carbon bonds needs to be as minimal as possible. Because imagine it like being on a Ferris wheel and trying to catch a penny, right? So molecules that are very short are going to have a much easier time holding on. They're going to get less sick than the ones that are further out. Because the molecules that are shorter, right, they have a very limited rotation. Whereas the molecules like beta alanine that have rotation about 2 bonds, it makes it harder for that molecule to react with some other molecular species because it's rotating more consistently And it's harder for there to be a stable chemical interaction between that amino acid and its and a given compound. So those are two ways in which it can be selected for. The third way, so like what you were mentioning, right?
How do you? There was no selection immutable. If there would be amino acids which are not functional, which cannot be used in the chain, they would also react and they would become incorporated, and then the chain would simply cease to get longer in order to have a chain of the size, which then would become a functional ribonuclease or whatever other protein. As you know, the average size of the proteome of the proteins in prokaryotes is about 300 amino acids.
Okay, and so here's the thing, right? You're partially correct, but let me change this to an analogy of being something like tug of war, right? Because it is true. You're going to have various protocell systems that are essentially undergoing these reactions and creating energetically favorable systems and potentially even replicating to some extent. However, the determining factor, and I also have to go pretty soon as well, is how quickly can this molecule produce energy? How quickly can it transform reactant A into product B? So molecules that are shorter, in the case of amino acids, molecules that have less rotation about carbon-carbon bonds, are going to catalyze those reactions much more quickly in comparison to amino acids that have more carbon separating their N-terminus from C-terminus. And because of that, once these systems begin replicating, you will naturally have systems that have incorporated more alpha amino acids compared to all other systems. And for that reason, it becomes predominated.
Well, in my understanding, immutable, what would happen, even let's suppose that you would have a miracle and amongst a sequence space of trillions and trillions of trillions of sequences which are not functional, that you would get to a first enzyme or ribonuclease, which in a fully fledged and functional cell would emerge on prebiotic Earth. Mm-hmm. It would just lay around there and then disintegrate again. Do you know why? Because proteins and enzymes, they have only function if they work in an integrated manner together with other proteins and enzymes as a system. It's the same situation as if you would have a piston just laying around somewhere. It would have no function. A piston has only function integrated in the motor block, in the car, in the whole system as a whole. Then it has function, but alone, separately, it has no function at all. And that's the same with proteins. If they would just lay around prebiotically, if they are not incorporated in a meaningful and functional manner in the cell, they would not have any function at all. They would just lay around and then disintegrate again. That's why I say that cells are irreducible. You need the functional whole all together all at once.
So again, and if we talk with Dmiter again, I think this might be an interesting topic of conversation. But I'll direct us back to when we were talking about the conversion of adenosine diphosphate to adenosine triphosphate, ADP to ATP. So one of the reasons I see that as a critical point, a really turning point in this conversation, is once you have an energy source, in this case ATP, but it could be anything else for that matter, it's not important what it is. Once you have an energy source, right, you have more energy that's available to a system, and that means that the system can do other things. It means that
the problem that I also see here, as you know, that ATP, for example, is used in a very precise manner in proteins at the right place where the reaction takes place. It isn't just energy somehow. It is the same as gasoline. If you have just gasoline, you incendiated that you have an explosion, so you need the gasoline at the right place where it can take its reaction, and that's the same in biological systems. It's not so that you just have ATP available. You need to have ATP directed in the reaction where in the enzymes the reaction is required and where it's used to perform to have the energy available. So, it's not just floating around ATPs.
So let me make this point, right? So it is true that some molecules use ADP and ATP as essentially regulatory controls. However, those systems are something that would evolve later on. The important aspect of the system is that once you have a source of energy, like ATP, the simple system, which consists of just protein, which consists of small peptides and nucleic acids, it is allowed to, it can evolve further. And the reason for that is, for instance, if the largest chain you can produce with just autocatalytic cycles is, say, 5 or 6 amino acids together, so a 6-chain peptide, right? Well, there's a limit to how much activity it can have just by virtue of what chemicals are interacting. However, when you have
I'm sorry, I was distracted. I had to answer my daughter.
No, it's fine. But the point is, if the smallest chain you can produce without any complicated energy source is, say, 6 amino acids in length, right? Then more energy available to the system is going to say...
There was a system yet. If you have a chain of 6 amino acids, that's not a system. That is just a small polypeptide.
Yes, but the point is that those small polypeptides, based upon their structure, have some capability to react or to have some function, be it storing
So do you think they would rather disintegrate or rather complexify? What do you think is the more probable trajectory if you have a certain kind of complexity here?
They will complexify to an extent.
To an extent. And do you think it would be plausible that it would complexify to an amino acid strand of 300 amino acids, or in case of helicase, 1,000 amino acids in the right sequence?
Yes, but under a very
amino acid.
So the answer, and I'll have to preface this, is yes, but it has to be very specific. And the reason I say that is, once you have an energy source that can begin connecting more things together, so in this case amino acids, right? What becomes more relevant is that these proteins have a specific place to live, right? So part of that energy, in my opinion...
There is no reason why they would live or exist in a specific region.
Yes.
We have
so the reason...
visualize there is just randomness. There is just natural elements. There is nothing to sort out or to provide a favorable environment or something like that.
No, but here's the thing, right? Is that proteins, as we were saying earlier, proteins have an affinity towards chemicals based upon their structure, right?
Mm-hmm.
Well, it might be that when you don't have a complicated energy source, you only have enough energy for that protein to do some chemical reaction, and that's it. But if you have an energy source, right, what might become possible is for that energy to be used to bring lipids or other systems which are more hydrophobic. So compounds that aren't that are interacting with other parts of the protein, that are interacting with the hydrophobic elements of the protein to protect it from its environment, so that this protein isn't undergoing random reactions elsewhere.
Do you see how you constantly try to bring purpose into your explanation when there actually at that stage there was no purpose? There was no purpose to encapsulate a protein into a lipid membrane or so. That there was no aim for that.
So the only aim that's being considered, right, is to increase entropy, is to increase the entropy of a system to its environment.
But natural forces, they do not have that urge to form an energy gradient. And it's not only the energy gradient, but as you probably know, you need a concentration of calcium about 10,000 times less inside of the cell than on the outside. And if you don't have that gradient, you cannot have signaling. And if you don't have signaling, you have no life.
Again, right? You're taking modern cells and dragging them back 4 billion years back. So the most basic thing that Dmiter is describing is that in the very first stages of life, and the other steps are more complicated, and I'll give it to you, that these things are complicated and we don't precisely know how those trajectories might work out. But what is clear is that once you have this type of system that's functioning, so very small amino acids and nucleic acids, and an energy source.
That's not the system. That is just...
It is.
Okay.
It's a system, guys, like Dmiter was saying, that's capable. So, like Dmiter was saying, it's a system that is capable of undergoing Darwinian evolution. It is a system that is capable of replicating its components.
I really disagree with trying to snake in Darwinian evolution when you don't have...
What in the hell is going on?
So, I have no clue. Is it bleeding over?
Who's having the party?
I'm using internet that's not my own, so I don't know why it's doing that.
What?
I'm in the middle of a mall, so if you hear other things, I apologize for that.
Oh, okay. I thought it was like a pool party. Anyway, okay. Well,
but so really briefly, can I ask you why you think that the definition of Darwinian evolution or life that Dmiter is proposing is incorrect or just not sustainable?
Well, because you need DNA replication in order to have allele frequency.
Oh my God, you didn't listen.
And where you can select from. That's basically where Darwinian evolution starts. There was no selection process in a prebiotic environment.
So,
as I said, chemicals, they do not have any urge to be selected. Also, there are just chemical reactions.
No, but and the point that's being missed, and why I encourage people to study other chemistry before they study cell biology and biochemistry, is chemicals have a desire to be stable. And if that means that other chemicals are interacting, so if that means that amino acids and peptides have a favorable interaction with each other, that's something that's going to occur because it stabilizes the molecular structure.
Okay.
Well, and the other part of it is that if that system can be replicated, right? So if this small little system can replicate and form more of itself, then it is, by definition, evolving. It is replicating its own information.
There was no information at that stage.
The information is within the stability of the complex, right? That's the type of thing Dmiter is proposing as the origin of the genetic code.
Yeah, I am very skeptic about the claim that you can have RNA replication. I wrote already, and I quoted why I don't believe it, and Conin points that out. So there are so many claims made which are unsupported and scenarios which are just hypothesized but which are not confirmed.
What do you mean unsupported? What do you mean unsupported? All of these papers have...
So, for instance, the Sutherland paper was brought up earlier, and what Dimitri pointed out, and I've read part of the Sutherland paper as well, is that Sutherland proposed initially a mechanism, and then experimentally later, he proposed that it actually was plausible.
So I am talking about RNA replication.
Yeah, that's exactly what the Sutherland paper was talking about.
Well, I can...
He was talking about... So the Sutherland paper that was being referenced was talking about how if you take a particular strand of RNA, under the right chemical circumstances, it can begin to polymerize to a specific length, so about like 8 to 10 nucleotides in length.
Okay.
That's what the paper was describing. So the claim about, so this whole claim about RNA not being able to replicate is just wrong. It's incorrect.
Well, show the paper. I'm ready, willing to.
The paper we were discussing earlier did exactly that.
Okay.
The paper on John Sutherland.
Yeah,
so,
I've not read. Well, the one that I quoted doesn't make that claim as far as I have read it.
But so let me ask you this then, right?
And it's not about self-replication. That paper is just about the attempt to make RNAs. That's a different topic.
Okay, but so part of what replication means is that the ability of RNA to polymerize, right?
Yes, but that paper was attempting to actually synthesize prebiotically RNAs, the monomers.
It's polymers.
So it's not about polymerization. It's just about making the RNA itself, the monomers.
If I'm recalling, I could be recalling incorrectly, but I believe the paper was about how those monomers would polymerize into small chains of oligomers.
Yeah, what I know about this,
But so the point I was mentioning is that if the paper does say that, and I'll read it again to make sure it does, right? If the claim is being made that it can't do it, but you haven't read the paper, right, in its entirety to make sure that that's correct, why are you making the claim, right? It'd be like me saying that something can't happen because this paper mentions a difficulty with it, but in the actual paper, it says the exact opposite.
Well, as I said, I have made a video on RNA synthesis prebiotically, and when I did the video, I mentioned that paper. I took some information from it. It was a couple of years ago or so. For what I remember, that paper was mentioning the attempt to make the RNAs, the monomers, not about polymerization. Since it is a couple of years ago, I can be wrong now, so that's a possibility.
Yeah, I would recommend we both, at this point, we've reached, at this point, kind of a standstill in the conversation, because Dmitry has kind of explained his own position, and now other issues that are coming up, like, for instance, how do you cause the DNA strand to, or RNA strand in this case, to polymerize, is an entirely different question.
Yes.
I remember something very specific about this Sutherland paper, and it seems like that from what we're describing, you're recalling something different.
Yeah, I have written an article on that specific topic, so I want to make it available to you and to others. I quote in there so you guys can read it. If there is newer information in regards of RNA polymerization, then I am, and the problem has been solved, then I am happy to admit it. I have no problem. But for what I know at that moment is that RNA polymerization or self-replication is impossible.
So, like I said, it seems that we are both reading the same paper, and that we are either misunderstanding certain aspects of the paper, or that one of us is talking about one part of the paper and the other part of the paper seems to disagree according to our recollection of it.
Yeah, we would have to read. I would have to read it to see if it talks about RNA replication. What I remember, it was talking about the prebiotic synthesis of RNA.
Right. So at this point, I would say that it's best to just stop the conversation and regroup. So look into this topic a little further, because at this point, it just seems that we need to do more reading into the topic before anything productive can be gained from it.
Well, I have quoted here. I put the link forward in regards of that topic. I have investigated this, but if there is new, more recent research and evidence which proves earlier conclusions, as I said, I am ready to accept it. I have no problem with it.
Right. At this point, we're just both. At this point, we're both pissing in the wind.
Yeah,
we don't recall what the papers say, and it's easier for us to read some of the articles and see what it actually states and then regroup.
We have to end it, guys. Otherwise, we're going to go over the transcript limit, and I hate that. So, so anyway, thank you, Otangelo, for showing up. Thank you, Immutable. Say hi to the people at the party that you're at.
I will try. I will try.
You know, we're looking. We have a place here in Minnesota, a homeless shelter. Not sure if you're interested, but
you know, Michael, I was hoping for the day you said you would adopt me, but I didn't know we were going to do it publicly.
Do you mow lawns and, you know, do you do windows?
I can teach you organic chemistry.
We kind of need a window washer. Okay, guys. Anyway, good having you, Otangelo. We're gonna do some videos about you, and you should do some videos about us. I just don't see where we're getting anywhere with talking to you, because you have an impassable gap. For one thing, I think you should learn some chemistry. It might help. I don't know. Anyway, but you've been very gracious, and thank you for showing up.
Okay, thank you guys. Thank you for having me at your channel, Mike, and you guys are always welcome to show up at mine when you want.
Yep, and thank you everybody in chat that has been watching. Thank you for the 9 likes, and fuck that guy that gave us the thumb down. I think it was standing for truth, probably. So anyway, we'll see you guys.
Okay, bye-bye.
Okay, we'll end the broadcast, and that'll be it. Goodbye.